Good morning, all. Good morning. Good morning. Good morning, Sunita. Good morning, Sadhguri, Sweta, Oya. Good morning, all. Okay, so as I told you yesterday, good morning, Saima. We are having two classes today. Okay, at 11:30 a.m. also we are going to continue the plant physiology secondary metabolites concept. Good morning, Priya. Good morning. Good morning, everyone. Okay, let's continue. So friends, in the last class, good morning, two Priyas are there. Good morning. In the last class, friends, we were studying about the DNA repair mechanisms. If you very quickly recall, friends, if everyone quickly recall, while studying the DNA repair mechanisms, good morning, Gurunalini, good morning, Lakshmi. We were studying first the types of damages. Okay, depurination, deamination, oxidation, alkylation, dimer formation, mutations caused by the base analogs, as well as friends, mutations caused by the intercalating agents. This is what we people actually reading. Good morning, Sumaya. Please continue. Friends, I'll quickly recall everything in 2 minutes. Good morning, Reens. Okay, so when we were reading the depurination, what did I say to all of you? Depurination is a process where there will be a removal of the base. How there will be a removal of the base occur, friends? By the cleaving of glycosidic bond. That is only, friends, we can call it as the apurination or depurination. Later we read, friends, deamination. I'm just quickly recalling everything. I hope every one of you remember about the deamination. What did I tell you about the deamination, friends? Where there is a removal of the amino group, where cytosine converted into uracil upon removal of the amine group. I told to all of you, friends, you have to remember all this without fail, because in the part B question, even we can also expect in the part C question also in the form of a matching. Deamination of which base is basically converted into what? So it's very important, you have to remember. Then 5-methyl cytosine converted into thymine, adenine converted into hypoxanthine, guanine basically converted into xanthine. Please try to remember. Later we studied what is oxidation also. What did I tell you about the oxidation? Good morning, Jayshree. The treating of the DNA with what, friends? The treating of the DNA with the oxidating agents, where the guanine, upon treating with many oxidizing agents, actually what happens here, this particular guanine, it is converted into 8-oxoguanine, where this particular oxo group was added at the 8th position. This is what we can call 8-oxoguanine. I also told to all of you, 8-oxoguanine will cause the powerful mutation which leads to the damage of the DNA very quickly when compared to the all other type of the repair mechanisms, all other type of repairs or damages. Alkylation, I hope all of you remember. So the treating of the DNA with the alkylating agent or the methylating agent completely changes its base conformation. If every one of you very quickly recall, we discussed, friends, cytosine upon treating with what, friends? Cytosine upon treating with the hydroxyl amine. All the students try to remember. This is the cytosine, right, friends? Cytosine having a NH2 group, we all know that. So friends, this particular cytosine, it is basically converted into what now? Hydroxyl aminocytosine. So friends, this particular hydroxyl aminocytosine is basically converted into what, friends? Instead of pairing, see, cytosine originally has to pair with guanine, we all people know that. But what's happening? Good morning, Ritika. But upon addition of the hydroxyl group to the amine of the cytosine, it is converted into hydroxyl aminocytosine. So friends, hydroxyl aminocytosine never ever pair with the guanine. Instead of pairing with the guanine, it's gonna pair with the adenine. As a result, friends, entire transition got changed from CG to TA transition. That's a very simple point it is. Now next, if you look into the guanine, what is the point you have to understand, friends? Guanine, whenever treated by methylating agent, what's the best methylating agent? We can call it as MMS, methyl methane sulfonate. So friends, what's happening here? Guanine at a particular position, which position friends? At a O6 position, it is added by the methyl group. So we can call it as what? O6 methyl guanine. So we all people know that guanine originally has to pair with the cytosine, but friends, this O6 methyl guanine, instead of pairing with the cytosine, it is paired with the adenine. Once again friends, the transition completely changed from GC to AT. One of the dangerous mutation, I already told you. So we exactly stopped in the last class, that is the dimer formation in the bases. What did I tell you about the dimer formation in the bases? I hope every one of you remember. Originally the base thymine has to pair with what friends? Adenine, right? Two thymine molecules originally has to pair with adenine, but everyone try to understand, look into the picture friends. Whenever thymine molecules are exposed to the high intensity of the UV light, what friends? Whenever thymine exposure to the high intensity of the UV light, immediately what is going to happen friends? There is a formation of the thymine dimer. We can also call it as what? Thymine dimer bridge. And I hope every student remember, in the last class I told you, this particular thymine dimer, it looks like a chemical. It's look like a chemical, cyclobutane. So that is the reason friends, we will be calling it as the cyclobutane photodimer. What do you call, my friends? We will be calling it as the cyclobutane photodimer. Please kindly don't forget it. Friends, if we don't break this bridge, everyone listen to me carefully. If we don't break this bridge and if we don't separate the two thymines, friends, it leads to a very dangerous skin cancers, which we can call melanoma. So what is this pyrimidine dimer? Two thymine molecules associated with each other, and they are going to form a particular bridge. That particular bridge only we can call it as the thymine dimer bridge. And what is the function of this particular thymine dimer? What happened? Instead of pairing with two adenines, they are pairing with each other. So if the same genetic information passes to the next generation, it becomes permanent and it causes very dangerous kind of mutations. So that's why we have to break this bridge and we have to separate the two thymines, and we have to allow the two thymines to pair with the adenine. That's a simple one. So friends, till here in the last class also we discussed. Now there are two more type of damages are there. Very important type of damages. See friends, 99% in a given 2-marker question, mostly they can ask the question from the type of damages only. Otherwise, in the 2-marker question, as a direct and straightforward, they can also ask the question from that particular, what we can say, that name of the repair mechanism also. Okay, anyway, look into this. Next one is friends, mutations caused by the base analogs. Okay, so everyone try to understand what is base analog. Understand friends, base analog means, don't confuse, they are structurally similar to normal base. All of you understand? They are structurally similar to the normal base. What about functionally friends? It may perform the same function or it may not perform the same function. We cannot conclude whether it is performing the same function or not. It may or may not. But friends, structurally they are exactly similar. Every one of you look into the picture very carefully friends. Don't get confused here. All of you understand. So friends, this is what? 5-bromouracil. What was that? 5-bromouracil. Friends, you know 5-bromouracil exactly behave like thymine. Exactly behave like what friends? It is exactly behaving like a thymine. every one of you listen my statements very carefully. Originally we know that thymine has to pair with the adenine, we know that. And what did I tell you? 5-bromouracil also behaving like a thymine. I told you one interesting point. What? When it comes to the function, it may perform the same function or it may not perform the same function. So here what I want you people to remember is that 5-bromouracil behaving like a thymine. So friends, it will go and pair with the adenine in the normal state. There is no problem. No problem at all. But now you look into the down. This is very important. This particular 5-bromouracil, it's a behave like a cytosine, in which state? Rare state. As a result, originally friends, 5-bromouracil behaves like a thymine, thymine pair with the adenine. But what is happening now? 5-bromouracil is not behaving like a thymine, friends. It's behaving like a cytosine. Then cytosine is basically paired with what, friends? Guanine. Entire base transition was changed. I can say, friends, this is also one of the very dangerous mutation. Please kindly do not confuse with this also. Next, another very important one which were asked in our exam also, that is mutation caused by the intercalating agents. Okay, everyone listen my words first carefully. What are intercalating agents? How can I define the word intercalating agents? Everyone listen, friends. This is 2 times directly asked part B question. The question is very simple. All of the following are the intercalating agents except. Okay, and they have also asked one time question, which of the following agents can cause the frame shift mutation? Once they asked a question, which of the following agents can cause the frame shift mutation? Second time they asked a question, all of the following are intercalating agents except. These are the 2 questions they have asked. And let me define you first, what are intercalating agents? Friends, intercalating agents are hydrophobic molecules. Every one of you listen to me very carefully, friends. You should not think that, listen to me, you should not think that some other enzymes or some other molecules, they will introduce the intercalating agents in the DNA. That is absolutely wrong statement. All the students listen to me. Intercalating agents on its own, once again listen to me, intercalating agents on its own, it can introduce it in the DNA. It will randomly chooses a certain basis. What, friends? It randomly chooses a certain base pairs and it will introduce in this particular DNA. So friends, what you people has to remember for the exam, it includes ethidium bromide, acridin orange, then friends followed by the actinomycin D. These 3 are very important ones. What are those? ETBR, ethidium bromide, acridin orange and actinomycin D. I want all of you remember, among these 3 also, friends, acridin orange is one of the most important intercalating agents. Please kindly do not forget this particular point at all. Now, all of you listen to me carefully. What this intercalating agent will do, friends? I told you now, they will cause great damage to the DNA. They can distort. Friends, here distort means damage. Continue, everyone. They can damage the DNA double helix. And friends, so you only try to understand, friends, if the DNA helix get damaged, how does the DNA replication occur? That's not even possible. So DNA replication never occur, DNA transcription never occur, and friends, even doing the DNA repair mechanisms are also a very complicated actually. Always remember that. And what is the unique statement you people has to remember? Listen to me carefully. Whenever an intercalating agent is introduced in the DNA, friends, it will cause either deletion or insertion. So basically deletion or insertion, what do we call, friends? Frame shift mutations. Please kindly do not forget this. Okay, now I want you to remember as it is, like what it is causing. Mutation by the addition, mutation by the deletion. Which means that, friends, let me tell you in a simple way. How the intercalating agents will cause the mutation, we will read in the genetics chapter. So genetics chapter also, there is a sub chapter called as mutation. So when we are reading that particular mutations, we will understand this elaboratively. All of you listen to me very very carefully, friends. This is what a molecule of intercalating agent. What you people has to understand, does this particular intercalating agent, do you think it has a particularly chosen a base and inserted over there? Absolutely wrong statement. This particular intercalating agent, friends, randomly it has chosen one base and introduced over there, right? And in case of a deletion, what happen? Whenever the intercalating agent introduced on at one particular area, suppose I have removed that intercalating agents. Imagine I have removed this particular intercalating agent during the process of replication. What happens, friends? There will be a complete deletion of the base. Friends, it's a common, right? All of you listen. In a coding sequence, suppose example, this is a coding sequence. CCU, CCA, AGC, CCA, like this many coding sequences are there. Because of the intercalating agent, one base got completely deleted, friends. Whenever there is a deletion of one base, what happen? Listen to me carefully, friends. Entire coding sequence is getting damaged. Entire coding sequence will get damaged immediately. Kindly do not forget that point. And one more very 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 important point, what I want you people to remember is that, now listen, here what I have done, instead of removing anything, I have added here. See here, after C, I have introduced a new base, CAC. Now CAC, UCC, AAG, CCCC, only A become left. A become left. So what you have to understand, once again, listen to me, either by addition or by the deletion, the entire DNA coding sequence is getting damaged, which leads to a serious damage. Because friends, whatever the protein that is getting synthesized, that protein either will become hyperactive protein in one case, in another case the protein will become completely non-functional protein itself. That is the reason, friends, intercalating agents generally cause the frame shift mutation either by addition of the nucleotide or our friends or by the deletion. Very very simple it is. So friends, so far what we discussed, I am telling you friends once again, DNA repair mechanism is very important concept. I hope you know that. This time we got in the February 2022 paper, we got a direct part C question. And does the question is very difficult? No, the question is very easy, but the statements are very tricky statement. Surprisingly, they did not ask the question about any enzymes. They did not ask the question about any mechanism of action also. Surprisingly, they asked a question between differences between one repair mechanism with the another repair mechanism. They just asked differences. So friends, right now we read all the type of the damages. Okay, this many damages are possible. Now if we leave the damages as it is, obviously what happen, it becomes mutations and it causes several genetic abnormalities. Even it causes several cancers also. Good morning Srikant, continue. Now friends, listen, how we are going to repair this particular damages. So friends, we are going to repair this particular damages by using several repair mechanisms. I always encourage you to remember the short names, friends. Mismatch repair, we can call it MMR. Base excision repair, BER. Always remember short names. NER, nucleotide excision repair. Next one, friends, direct repair. Friends, please kindly don't confuse, okay? The direct repair mechanism, friends, we will be also calling it as photoreactivation. Please kindly do not forget the statement at all. Next one is what, friends? Recombination repair. And the last and final one, SOS repair. So friends, SOS repair can also call it as error prone repair. What do you call, friends? We call it as the error prone repair. So friends, let me tell you one very interesting point here. All of you just listen to me very carefully. In case of a mismatch repair, BER repair, nucleotide excision repair, recombination repair and SOS repair, everyone listen my words. Whatever the part, listen to me, whatever the part of the DNA that get damaged, we remove that particular damaged part, we replace that particular part. See, when we have a bike damage, if a particular part of the bike got damaged, we will remove that particular part and we will replace with the new part. Same thing will happen in these 5 repair mechanisms. Whatever the base that got damaged, we will repair that particular, we will remove that particular base, or we will remove that particular nucleotide and we will replace with the new one. That's a no problem. But friends, in case of, there is only one exception, that is direct repair or photoreactivation. Friends, what is the speciality in case of a direct repair or photoreactivation? Friends, we don't remove, listen friends, this is really important statement, we don't remove the damaged part. Instead of removing the damaged part, we will repair there itself, and we will repair there itself without removing any damaged part. That's what we can call a direct repair. An example, in my bike there is a brake problem or there is a clutch problem, so I am not removing the clutch and I am not replacing with the new clutch or a new brake. What I am doing, by there itself I am repairing that particular brake without removing it. Same way, whenever there is a base got damaged, what we are doing, we are not removing it, we are repairing there itself. That is the reason, my friends, we will be calling this as the direct repair. Do not forget this particular point. So friends, now listen. You can ask me, sir, what exactly we have to read in case of a repair mechanism? How we can expect the questions? So friends, please every one of you listen to me carefully. In every repair mechanism, what exactly you have to remember? What kind of damage can be repaired by the repair mechanism? First point. What kind of damage can be repaired by the repair mechanism? First point. Second, and the most important point, what are the enzymes involved in the repair mechanism? Second point. Come on friends, be active. Third, the most important one, we have to remember the mechanism of action. How does that particular enzymes are showing their activity? So friends, only these 3 we can expect the questions, not in any other manner. Okay, only these 3. Which means which repair mechanism is used for which damage? What are the enzymes involved in every repair mechanism? How they are basically acting on? Simple point. So now let's talk one by one. So friends, how I will teach you is, listen to me very carefully. How I will teach you is, first I will explain one repair mechanism briefly, then I will show you an animation, then automatically you will understand. See friends, watching the animation will help us to visualize that concept. By that our brain can remember for the longer time. So whenever you come across with any kind of the animation about a particular process or a particular mechanism, always try to remember in the form of a visualization. Brain will catch it very faster when compared to normal reading. Okay, now listen to me very carefully friends, all of you. Give me 5 minutes, I will explain it briefly. Mismatch repair. Okay, Friends, while studying the process of the DNA replication, I told to all of you many times. What did I tell to all of you many times? DNA polymerase is the most versatile and accurate enzyme. I told you. Yes or no? Of course, yes. And what did I tell you, friends? DNA polymerase enzyme just will make one mistake. That's all. DNA polymerase enzyme makes one mistake out of 10 power 6 nucleotides added, which means, in a simple words, out of 10 lakhs of nucleotides added, DNA polymerase enzyme will make only one mistake. But friends, of course, even though it made only one mistake, but friends, suppose if that particular mistake is in the coding sequence, listen my words very carefully. Even though DNA polymerase enzyme made one mistake, even if that particular mistake is in the coding sequence, the entire DNA sequence will be altered, the entire process will be stopped, which leads to very powerful mutation. Even though one base got damaged, even though one base got mismatched, we have to repair that compulsory. An example, my friends, an example. C could be inserted opposite of an A. You all know very well, adenine has to be paired with the thymine. But instead of pairing with the thymine, adenine is pairing with this cytosine. Right? Because of this, what is happening? Entire transition is going to get changed. Suppose example, this is the sequence. G, G, C, C, C. This is the sequence. Originally G has to pair with C, C has to pair with G, and G, G, G. This is the original sequence. You know what happens sometime? In between this, there is an introduction of A, A, A. So, but see here, there, once again I will write here, just look into this. Originally, this is the sequence. G, G, C, C, C. So, C pair C. This should be the pairing. But you know what happens? Sometimes what happens? There is an introduction of the bases in between the other bases. See here. But in the opposite strand, what is there? In opposite strand, G pair with C, G pair with C, C pair with G, C pair with G, C pair with G. Then what about the A? It did not pair with anyone. If it is not pairing with anyone, what is happening? Obviously, it creates a gap. Now, if the same genetic information passes to the next generation, friends, undoubtedly, it will cause the mutation. So, we have to repair this particular damage. How we are going to repair this particular damages? By a repair called as a mismatch repair mechanism. And every student has to remember one common point. What is that? One common point is that this mismatch repair mechanism is depending on the principle. What do you call that particular principle, friends? We call that particular principle as the methylation. What do you call that principle? Methylation. That's why we can call it as methyl mismatch repair. I will explain simple way. Please listen to me carefully. If you listen to me clearly, you will get the concept clearly. See friends, there is a parent strand. This is the parent strand. Okay, first let me draw here, then you people will understand. Listen to me. These two are parent strands. Okay. Friends, you all people know, right? On the DNA, palindromic sequences are very common. Most of the students will think that, most of the people will think that palindromic sequences are not present on the DNA very commonly or very oftenly. They think that palindromic sequences are present very rarely. No. I want you to remember, friends, every 1,000 nucleotides, at least a few palindromic sequences are compulsory present in this particular DNA. So friends, suppose here there is one palindromic sequence and there is one palindromic sequence. So these are all what parent strands I am talking about. So friends, this particular palindromic sequences got methylated. So every one of you, please kindly look into the diagram, friends. This is what? palindromic sequences present on the parent DNA, it got methylated. Now all the students listen to me. This is the parent strand and this is the daughter strand. Everyone listen to me. Friends, all of you listen. Where is the damage now? The damage is here. Here in the sense what? The damage is on the daughter strand. Now we have to remove this particular damaged area. Listen to me friends. We have to remove this damaged area and we have to replace with the new area. That's a very simple point. Now every one of you listen. If we want to repair the daughter strand, listen to me carefully. If we want to listen to me, if we want to repair the daughter strand, first of all the mismatch repair mechanism enzyme has to differentiate. What they have to be differentiated? They have to differentiate which is the parent strand, which is the daughter strand. If they don't differentiate, they can't do the repair. Simple point. So now the question is here, how they are getting differentiated. Every student try to understand friends. This is what strand? Parent strand. Parent strand having palindromic sequences, it got methylated. Friends, you should not think that daughter strand is not getting the methylated sequences. Sorry, palindromic sequences. Undoubtedly, daughter strand also having the palindromic sequences, no doubt about it. But they are not yet methylated. Once again, listen to me. They are not yet methylated. When they are not yet methylated, you know friends, what do we call them as? We will be calling them as the hemimethylated DNA. What do you call friends? Hemimethylated DNA. This hemimethylated DNA, after certain time, it is also converted into methylated DNA. Now you can ask me, sir, you said that hemimethylated DNA, how it is going to be converted into methylated DNA after some time? With the help of a powerful enzyme. This is also asked a direct part B question. We can call it as a DAM methyl transferase enzyme. Deoxyamino methyl transferase enzyme. This is the enzyme which will add the methyl group to the palindromic sequences present in the daughter DNA. Why I am saying to all of you, every student listen this point. Whenever the daughter DNA is in the hemimethylated form, we have to repair the damage. Once again, I am repeating my statement. Whenever the daughter DNA is in the hemimethylated form, during that time only we have to repair the damage. Sir, why like that? Because friends, after some time, the daughter DNA which is hemimethylated, it is also converted into methylated. Then friends, how the DNA repair mechanism enzymes can differentiate which is the parent strand, which is the daughter strand? They can't understand. If they can't differentiate, they never do the repair. Simple point. So in a simple way, we have to repair the damage in the daughter strand whenever the daughter strand is in the hemimethylated form only. Every one of you listen to me carefully. There are different type of repair mechanism enzymes are there in this particular mismatch repair. Among those, the three major enzymes, just listen to me carefully. Once you see the animation, it will be very clear. Mut-H, Mut-L, Mut-S. Friends, why we have given the name mute? We called it as mute because friends, they all are derived from where? They are all derived from the mutated E. coli. That is the reason, my friends, we will be calling this as what? Mut-H, Mut-L, Mut-S. Every one of you, please kindly listen to me carefully. So first, what is going to happen? First, friends, Mut-S. Which one? Mut-S. It will start scanning the DNA. Listen to me carefully. What happens first? First, Mut-S will keep on scanning the DNA. Once it recognizes, suppose example, this is the damaged area. Once it recognizes the damaged area, what is happening? Immediately, friends, Mut-S recruits the Mut-L. Very simple point. Mut-S, once it recognizes the damage, it will recruit the Mut-L. You know friends, now Mut-S and Mut-L, suppose example, DNA is like this, correct na? The mut-S and mut-L make the DNA bent like this. See, originally DNA is like this, my friends, and the DNA is bending like this. Why? For easy cleavage. Now the point is here, the most important point. I have already told you, if the repair mechanism enzymes wants to be active, first they have to be differentiate or discriminate between the daughter strand and parent strand. Who will do that? Mut-H. What is the function of a mut-H? Mut-H actually will go and recognize the methylated sequences present on the parent strand. Okay, now mut-H recognized which is the parent strand. Mut-S and mut-L recognized the damage on the daughter strand also. Then immediately what happened? Mut-S and mut-L, friends, they are going to recruit a powerful enzyme exonuclease 7 or REC-J. Friends, what is that particular exonuclease 7 or REC-J will do? They remove all these damaged bases. They remove all these damaged bases. Now, once we remove all the damaged bases, you can see here there is a lot of gap got created. Friends, that gap has to be filled with the nucleotides. And friends, I am requesting all of you, please kindly do not confuse here. The enzyme comes here is only DNA polymerase 1, not the 3. DNA polymerase 1 will come and replace that particular gap with the nucleotide. Then ligase enzyme will come and seal the nick. But every student listen to me carefully. Suppose example, the number of bases that undergone the mismatch or are mismatching bases are more in number. An example, 10 bases got mismatched or 8 bases got mismatched. Then obviously Pol 3 only will come. If 1 or 2 bases got mismatched, Pol 1 will come. But 99.99% friends, Pol 1 is the only enzyme will remove this particular, will replace this particular nucleotides. That is what repair? Mismatch repair mechanism. So friends, now listen, I will show the animation. Once the animation is over, please give me in the chat box, please give me the reply that you understand or not. Say yes or no. Okay, just to give me one moment of time. Friends, understand it very carefully. Please kindly don't get confused. It's really good animation. Will explain us neatly.
When DNA polymerase replicates DNA, it uses proofreading functions to prevent the misincorporation of bases. However, DNA polymerase does incorporate an incorrect base for approximately 1 in a million bases synthesized, resulting in a mismatch that distorts the structure of the double helix. Curiously, far fewer than 1 in 10 to the 6 mutations actually arise. The mutation rate in an E. coli cell is only 1 for every 10 to the 9 or 10 to the 10 bases replicated. The cell has a number of repair methods, including one called methyl mismatch repair to remove and replace a mismatched base. Methyl mismatch repair involves recognition of the methylation pattern in DNA bases. E. coli methylates its DNA at the palindromic sequence GATC using an enzyme called deoxyadenosine methylase or DAM methylase. DAM methylase adds a methyl group to the adenine in both strands of the sequence. Adenine and thymine bases pair normally. The DAM methylase acts soon, but not immediately, after replication of a DNA sequence. Where DNA has methylation sequences, the parental strand will be methylated, but the new strand will still be unmethylated. The difference in methylation allows the cell to distinguish the correct sequence, presumably on the parental strand, from the incorrect sequence on the new strand. A series of proteins called mut-S, mut-L, and mut-H act together to repair the mismatch. Mut S first binds to the mismatch and recruits Mut L and Mut H. Mut L recognizes the methylated strand and brings it in a loop. Mut H then cleaves the unmethylated strand containing the mutation near the GATC. A DNA helicase called UVRD then unwinds the cleaved strand, exposing it to a variety of exonucleases. The result is a gap that is filled in by DNA polymerase I. The DNA polymerase continues through the region that originally contained the mismatch, inserting a correct nucleotide this time. After a while, the DNA polymerase falls off the DNA, and the gap that remains is sealed by DNA ligase. The methyl-directed mismatch repair proteins and genes are called mut because strains that are defective in one of these proteins have a high mutation.
Okay. So friends, is it clear? Everyone just give the reply once you understand or not. Hope it's clear for everyone. Please let me know, friends. If anyone having any kind of a doubt in the mismatch repair mechanism, I will re-explain. Please kindly let me know, everyone. Please respond. Okay, that's amazing. That's amazing. So friends, now the next type of the repair mechanism that we have to study is the base excision repair mechanism. So this is the question which were asked in the 2022 February. Okay, listen, this is the question asked and it's very easy question also. Listen to me very carefully first of all. Base excision repair. All the students, please listen my statements very carefully. What kind of damages we can repair here? We can repair the damages which occurred because of the process of deamination. See friends, I told to all of you very carefully, because of the process of the deamination, many wrong bases are involved. Yes or no? Of course, I told you, right? Because of the deamination, we already discussed, right? Uracil can formed, xanthine can formed, hypoxanthine can formed. What will form? Uracil, xanthine and hypoxanthine. Every student, please kindly listen to me very carefully. Friends, among all these bases, among all the unwanted bases, which is the base will form very commonly in the DNA. If I ask you a question, very simple question I asked you, which unwanted base, in other words, which unnatural base commonly found in the DNA? You people undoubtedly give the answer as uracil. Uracil is the unwanted base or unnatural base most commonly formed in the DNA. Please kindly do not forget the statement at all. Okay, now friends, listen to me carefully. BER, base excision repair, it can be initiated by an enzyme called as DNA glycosylase. Don't forget this point. If you remember the point of DNA glycosylase, half of the concept is very clear. DNA glycosylases are the enzyme which will cut the glycosidic bond. Now you have to ask me very quickly, sir, why we have to cut the glycosidic bond? Please tell me. See, because as I told you just now very clearly, there is an unwanted base is introduced in the DNA. Unnatural base is introduced in the DNA. I am requesting all of you try to understand the concept very clearly. This is the sugar and the first carbon of the sugar can be connected with the base. Yes or no? Either purine or pyrimidine. Then followed by the phosphate. Originally, what is our bases? Adenine, thymine, guanine, cytosine. Now, any of these four bases is not formed, friends. What base is formed? An example I am talking about. Uracil is formed. Now, you only tell me, do you think Is there any necessity of the uracil in the DNA molecule? Not at all. So what should I do? I have to remove this particular uracil, my friends. How I am going to remove that? That's the question. How I am going to remove this? By cleaving N-glycosidic bond. By cleaving what bond, my friends? By cleaving the N-glycosidic bond. Why only N-glycosidic bond? Because N-glycosidic bond is the one which connect the sugar with the bases. So that's why I will use the word DNA glycosylages. Now whatever the statement I am saying you is very important statement. The statement is DNA glycosylages are specific in nature. What do you mean by that? It depending on what kind of base is introduced, we will use a specific type of the DNA glycosylase. An example, uracil is introduced in the DNA. Then friends, what kind of enzyme we will use? We will use uracil DNA glycosylase only we will use, because this enzyme only will cut the uracil. Simple point. Suppose example, in the DNA xanthin is introduced, then friends, undoubtedly we are going to use xanthin DNA glycosylases only. Please kindly do not forget the statement at all. Uracil DNA glycosylase is introduced when the uracil is introduced. Xanthin DNA glycosylase comes into the picture. You will get to know, friends, whenever there is a xanthin is introduced. Very, very simple point. So what I would like to conclude, whenever there is a unnatural basis, listen to me, whenever there is a unnatural basis introduced in our DNA, to remove that unwanted or unnatural basis, we are using DNA glycosylases enzyme. This is the first and major enzyme. Second major enzyme, see friends, once again, every one of you please quickly try to understand. This is the base and sugar, and this is the base and this is the phosphate. What we have cleaved so far? We cleaved this, correct, right? After cleaving the base, we will cut this phosphodiester bond also. After cleaving the phosphodiester bond, we will replace the entire nucleotide. Simple point. Now, after cleaving the base, there is a no base, right? Then what do you call it? Epurinic site, epirimidinic site. You can also call it, friends, a basic site. A means no base or no purine or no pyrimidine. Very, very simple point. So friends, what is the second unique enzyme? You will get to know very easily. Listen to me. The second enzyme we called as AP endonuclease enzymes. What is this AP endonuclease enzymes are going to do? Friends, they are going to cleave the phosphodiester bond. Simple point, friends. Glycosylase enzymes are going to cut the glycosidic bond. Endonuclease enzymes are going to cut the phosphodiester bond. Now let me tell you in a simple and effective way. All the students pay utmost attention. Friends, focus. You can see right here, whatever I am marking here, there is an unwanted or unnatural base occurred. Let's take an example, uracil. Uracil is the unnatural base introduced here. What should we do now? We have to remove this. If I want to remove this particular base, first of all, what bond I have to cleave? I have to cleave the glycosidic bond. So friends, since uracil is introduced, which kind of enzyme comes into the picture now? Uracil DNA glycosylase comes into the picture. Very simple point. Friends, now this particular uracil DNA glycosylase enzyme cut the uracil. After cleaving the uracil, see here, every one of you look into the picture, my friends. Uracil came out. Now all the students listen to me. This is what bond now? Phosphodiester bond. I have to remove that also. Because once I remove the base, it create a AP site. We can call it a purinic site, a pyrimidinic site, or the abasic site. Then what does it create? It create a gap. So friends, in this particular gap, what kind of enzyme we are going to this site can be cleaved by what? AP endonuclease. AP endonuclease enzyme will come and cut the phosphodiester bond. Now everything can get removed out. This entire nucleotide is removed out. Now friends, the DNA polymerase enzyme will come and replace this particular gap. Finally friends, ligase is the enzyme will seal the nick. So now I am going to show the animation. Once the animation is over, please get back to me whether you understand or not. Please say yes or no. Always remember these things in terms of animation only. Okay, it's a very good animation. Try to understand.
About me? Let's see. Always a different.
Focus everyone, focus.
Some damaged DNA bases are repaired by a system called base excision repair. Base excision
Okay,
repair
I think voice is low. I will give the voice. Listen. See friends, there is a introduction of the uracil here. Correct na? So when there is a, this is nucleotide. This is entirely different. And friends, let me tell you one simple question. In our 2022 February exam, this is the question we got in the exam, exact question. The differences between the BER repair and the differences between the nucleotide excision repair. This is the exact question which was appeared. Listen, in the base excision repair mechanism, we will hardly remove one or two bases or one or two nucleotide. But in case of a nucleotide excision repair mechanism, we will remove bulk amount of the nucleotide. That's why nucleotide excision repair called as bulky repair.
or other defect to replace it with new DNA. Base excision repair acts on other kinds of defective DNA. For example, certain bases that are not supposed to be in DNA, such as uracil, are removed by base excision repair. If the base cytosine spontaneously deaminates, it forms uracil.
See, all of you look into this, we already read. Cytosine is converted into uracil because of what friends? Because of the deamination.
Incorporated erroneously into a strand during DNA.
So now we have to replace this particular
if
uracil.
in the place of cytosine, it would have a mutagenic effect. Whereas cytosine forms a base pair with guanine.
See, originally cytosine has to pair with the guanine, and see here entire base transition got completely changed out. So we have to remove this particular uracil compulsory in the DNA. Not only that, we already read, right, adenine is going to be converted into hypoxanthine.
For example, when adenine becomes
See, when there is a removal of the amine group from the adenine, adenine is going to be converted into hypoxanthine. That is what we read in the damages itself.
converting adenine to 3-methyladenine.
Now we have to remove that hypoxanthine also compulsory. Adenine originally paired with the thymine, but hypoxanthine, see friends, it will go and pair with the cytosine. Then the entire base transition will be
When it is
changed.
encountered in the replication fork, it stalls DNA synthesis. Consequently, repairing these mutations is vital for survival. Base excision repair.
Now all of you look into the picture friends. All of you look into this. How we are going to repair this particular base excision repair mechanism. How we are going to perform the base excision repair. First one, recognizing the damage. Who will recognize the damage? DNA glycosylase enzyme. Cut glycosidic bond. AP endonuclease enzyme. Cut phosphodiester bond. Then finally friends, polymerase enzyme will come and replace the gap. All of you look into
A
that.
battery of glycosylase enzymes that can recognize and clip damaged bases from the phosphodiester backbone. Hypoxanthine DNA.
So friends, listen, in the animation they said hypoxanthine DNA glycosylase. Why? Because it is cleaving the hypoxanthine only. If they said uracil DNA glycosylase, then it is only cleaving the uracil.
3-methyladenine.
See, this is called 3-methyladenine DNA glycosylase because it is cleaving only 3-methyladenine.
And uracil
Uracil DNA glycosylase is cleaving only uracil. So DNA glycosylases are specific in
These
nature.
bases are present in DNA. The glycosylases cleave
See,
the
see,
bond
everyone look into that.
damaged base
It
to
will
the
go and
empty
cleave that
ribose
base.
in the phosphodiester
Simple
backbone.
point.
The
Now,
result is an intact
now see here.
phosphodiester.
Now every one of you look into this. There is a gap. This is what do we call a purinic site, a basic site, a pyrimidinic site. Now this can be cleaved. The phosphodiester bond can be cleaved by AP
Missing the
endonuclease
base. The site is called an AP site.
or a
In this
purinic
case, it
site.
is missing a purine and is therefore an apurinic site. If it were missing a pyrimidine, it would be an
A pyrimidinic site.
The next step in base excision repair involves AP endonuclease.
Now look into this picture, the powerful enzyme, AP endonuclease enzyme. What is gonna cleave that? It's gonna cleave exactly the backbone. What's the backbone of the DNA? Phosphodiester bond.
That specifically cleave the phosphodiester backbone at AP sites.
See here, now my friends, there is a gap, but every student has to understand the wonderful point. Still the OH group is present. That's an important point, right friends? When the OH group is there, we can easily add another nucleotide and we can easily form the phosphodiester bond. Now who's gonna come into the picture? DNA polymerase 1.
DNA polymerase 1 to start adding nucleotides.
So it will start adding the nucleotide. Ligase will come and seal the nick. So that is about base excision repair mechanism. So depending on the type of base introduced, different type of glycosylages will come into the picture. I hope it's clear for everyone. If anyone having any doubt till the base excision repair mechanism, you can ask me. I will re-explain, please. Okay. I hope it's clear for everyone, friends. That's all for now, and I'm requesting all of you attend 11:30 a.m. class today without fail, because friends, secondary metabolites topic is extremely important in the plant physiology. If you look into 2022 February paper and if you look into 2020 November paper also, we got every time 6 marker, 1 4 marker and 1 2 marker question. So don't miss the 11:30 a.m. class, attend it without fail. Okay, that's all for now, friends. Okay, I'll see you in the 11:30 a.m. class. Bye bye. Thank you all of you. Thank you so much. I'll see you at 11:30 a.m. sharp.